All right. Welcome to the Friday UTIG seminar. I'm Peter Flemings. I'm pleased to introduce our speaker. And our speaker is Nicholas Tanner Mills, who goes by the name of Tanner. Tanner received his BS at Brigham Young, his MS at Baylor, and his PhD at Texas A&M. At Texas A&M, he studied under Julia Reese, whom you may remember as a graduate student here at UTIG. His MS work focused on paleoceanographic studies and accumulation of organic matter in middle Pennsylvanian strata in the Paradox Basin. And his doctoral work studied the interaction between microorganisms and clay diagenesis. I might stop here and say you might wonder why Tanner and I are studying permafrost in Alaska. And I would like to draw a couple connections, which is that Tanner's got deep expertise in microbial science from his work at Texas A&M. And as you know, I'm interested in hydrate systems. Turns out permafrosts are a perfect example of a system where we are consuming organic matter to generate greenhouse gases, and we're transporting fluids through an ice phase, which is not hydrate here, but ice. And so for us, it's a really neat step off into a new direction that still builds on some of what we've been thinking about over the years. Okay, with that, I did want to also point out Riley Garrett, who's a sort of co-investigator in the project, a graduate student within UTIG, and predicting greenhouse gas fluxes. One other comment, Alejandro, where are you? We'll be hosting lunch afterwards with the speaker, and I'll let him talk afterwards. Thanks, Tanner.
All right, thank you, Peter. Everybody hear me? Yeah, okay. My elementary school teachers told me my voice travels, so I'll trust them. So yeah, today I'm going to talk about trying to predict greenhouse gas fluxes to the atmosphere from thawing permafrost. The key word there is trying, and so this is a little bit preliminary, so I'm going to talk a lot about what we are doing and some of our preliminary results. And so as Peter said, he's, the methane hydrates guru and something this department knows a lot about. But on the other side, we have permafrost. And so there's a lot of similarities there. So there's porous media filled with ice-like substances or ice, microbial methane production, and it's a major component of the carbon cycle. And so this research I'm going to be talking about today is in collaboration with Sandia National Lab as part of the CH4 PACE project. And so we've got Peter and Riley and I here at UT Austin, and then we have collaborators at Sandia, the University of Alaska at Fairbanks, and Tufts. And special thanks for funding from UTIG and Sandia. And with that, a lot of us know, or you may know, that global temperatures are on the rise, and anybody that lived in Austin last summer might anecdotally know that this is true, but there's also data to back that up. And so here on this plot, we have a difference from a mean average. And so that average there is the 20th century average. And on the x-axis, we have years. And so we can see that before 1940, we were cooler than average. And since about 1980, our temperatures have been going up compared to that global average. And so this is at a rate of about 0.2 to 0.3 degrees Celsius per decade. And this seems very like a tiny change, but it's actually quite a great change. And this increase in temperature is coincident with global CO2 in the atmosphere. And so that's over here on the far side on the y-axis is ppm CO2. And so we can see this CO2 roughly takes an upturn when our temperatures are also going up. And so these are very linked together. And in the Arctic, this global warming trend is actually amplified, and it's about 2 to 4 times faster than the global average. And so on this plot here again, we have a temperature anomaly, or the temperature change from an average versus our years on the x-axis. And you can see in this really faint kind of grayed out data in the background is our global average temperature rise. And in the Arctic is these darker colors. And we can see that this rise in temperature is much steeper and is occurring much faster in the Arctic. And this is actually a problem for Arctic permafrost. And so as temperatures rise, the permafrost can thaw, and this permafrost has a lot of carbon that can be released to our soil and atmosphere system. And so if we look at this map right here, we're looking down on the North Pole with North America to the left and Russia to the right. And all the areas that are colored represent areas of the Arctic that are dominated by permafrost or have permafrost present. And then the different colors represent how much organic carbon is stored in that permafrost. And we can see with these orange and red colors that a lot of the Arctic actually has really, really high amounts of organic carbon that's trapped in these permafrost soils. And so these elevated temperatures are threatening to release this carbon, which can then be utilized by microbes and emitted up into the atmosphere. Now, if we take more of a zoomed in look at what a soil profile might look like in the Arctic, we could imagine a current soil profile shown here on the left and a future soil profile over on the right. And this very top layer of the soil there is called the active layer. And this is very obviously named, but it's active as in the summer, this layer thaws out, and in the winter, the layer freezes again. And below that, we might have a layer called a talic, and this is a layer that remains unfrozen year-round. So no matter what's going on up at the surface, it could be winter and there's a frozen active layer above it, but this talic layer will stay unfrozen year-round. And then below that, we have stable permafrost, defined as frozen for longer than 2 years straight. And we could theorize that as global temperatures go up, this active layer is going to get deeper and our talic layer is going to expand. And when this happens, this organic carbon that's trapped in the permafrost is going to become unfrozen year-round. And we can say that microbial respiration of organic matter might be increased with these warmer temperatures and the thawed soil. And so we can kind of look at what microbial pathways might be happening in these soils. And so up at the top of the section, we might have an oxic zone where there's oxygen present, and below that an anoxic zone where there's no oxygen. So up in the oxic zone, we might have aerobic respiration where this CH2O molecule, so that's representative of organic carbon, where microbes will utilize it as an energy source and reduce this oxygen, and they'll create CO2. Down in the anoxic zone, we might have these processes, so fermentation, where we take the organic carbon and we split it and make CO2 and hydrogen, in which case these hydrogenophotropic methanogens can utilize the hydrogen and CO2, and they'll make methane. And acetoclastic methanogens, they'll take acetate and split it and make methane and CO2. And so these are just some examples of microbial pathways that are producing greenhouse gases in the soils. And then these gases can make their way to the atmosphere. But one of our big questions here is, at what rates are these gases going to flow up towards the atmosphere? And so essentially this is a permeability problem, right? And so if we kind of think conceptually about the permeability or how flow in these sediments may be occurring, we can make a little schematic diagram where our sediments are brown, water is blue, ice is light blue, the gas phase is red, and we've got a little arrow for flow. So up in our active layer, in the summer, we might have two phases, gas and water, where the gas is flowing up through that pore. But in the winter, we might have three phases, where we have gas trying to flow, but we have ice that's formed inside our pore, and we might have a tiny, tiny bit of liquid water coating the grains as a wetting phase. And now if we look at the talic, this is going to be very similar to our summer in the active layer, and in our permafrost, it's going to be similar to our winter in the active layer. Now, one of the main reasons why we're doing this is because what's known about gas flow in permafrost systems is very limited, and the rates at which microbes can generate greenhouse gases and how they may do that with warming soils in the future are relatively unknown, and they're poorly constrained. And not only that, they're typically not included in permafrost thaw or global climate models. And so we have a lot of global climate models that are trying to say, we should have this much carbon in the atmosphere in the year 2100. And so the temperatures might be this, right? But they're not taking into account this permafrost thaw feedback that might be putting methane and CO2 up into the atmosphere. And so our aim is to integrate the permafrost flow and the source of that permafrost greenhouse gas emissions, and to use those together to predict greenhouse gas fluxes to the atmosphere. And so what we do in our labs is ultimately going to be used to provide flow and source terms for greenhouse gas global climate models. And so this effort is going to be spearheaded by our friends at Sandia. And so they're going to take our source terms and flow terms and start building some site-specific one-dimensional models. And then they're going to upscale those into regional to global carbon models. And so just an example here on the left, we have a 1D model where they've modeled temperature, ice saturation, CO2 production, and methane production in the sediments. And then it's coupled with the flow towards the surface. And then they'll be able to take these and say, we know a lot about the ecotypes throughout the Arctic, and then be able to predict what ecotypes may have what kind of flow properties in order to make bigger and broader models. And so with that, and trying to build towards that, this is what I'm going to mainly be talking about today. So we'll have our site and sediment characterization, some permafrost gas flow, we'll talk about that, and then gas production in permafrost. And so I'll start with this site and sediment characterization. And so our goals here are to collect core and characterize the sediments there, things like water content, grain size, grain density, organic matter, mineralogy. And essentially, these sediment properties are going to play a big part in greenhouse gas production and in sediment permeability. And so we identified two study sites up in Fairbanks, Alaska, which is kind of shown in this indent map right here. And then at the bottom of this map is the University of Alaska Fairbanks campus. And our sites are actually out on their cross-country ski paths during the winter. So it's kind of fun. They will yell at you if you park a snowmobile on the path. We've learned. But our two sites are Smith Lake #1 and Smith Lake #4. And yes, they're right next to Smith Lake. And so these two sites are actually quite different. Smith Lake 1 is in kind of some denser treed area where there's a lot of mixed spruce and moss. And Smith Lake 4 is actually out in the open with, I guess, some black spruces and some tussocks, but generally it's out in the open. And these two sites have been pretty well characterized in the past. And one of those things that have been characterized really well is ground temperature. And so there have been ground sensor temperature data being collected at both of these sites. At site Smith Lake 1 for about 20 years, and the other site about 10 or 15. And so as we look at this plot on the right, this is showing the yearly temperature average with temperature on our x-axis and depth on the y-axis. And so we can see some pretty stark trends here as the depth profile for each year is shown, with the purples being farther back in time, up to the yellows being more recent. And so there's a clear trend here of purples to magentas to yellows and oranges to the right as we're starting to move into higher temperatures. And so this is actually a stark example of permafrost ground temperatures rising or permafrost thawing. However, the permafrost here is not totally thawed. However, down deeper in the permafrost, it's pretty close to our 0 temperature in Celsius. And so it's kind of on the verge of being susceptible to thaw. And it's thawed about, I guess, 1 degree C deeper in the permafrost over the last 20 years. And so this is pretty drastic. At Smith Lake 4, we have our same plot with temperature up on the x-axis and our depth trend on the y. And from 2015, we have our dark purple up until 2022 in our lighter orange, yellows colors. However, at this site, we don't see quite the abrupt trend. We can maybe see a little warming, but it kind of bounces back and forth if you look close. And so here at this site, this is one of the colder sites in the Fairbanks area. And here we have stable permafrost. And so there's these two kind of different temperature regimes here that are really interesting. And so we went up to Alaska and we utilized a Talon drill system, which was fun to use. And so that's shown here on this picture on the left. And so it's kind of a tower tripod that has a DeWalt drill. It's kind of like a jackhammer, really. And so I've seen some construction workers in town using these to like jackhammer cement. And so then there's the drill pipe right down here. And so this is a master's student, Riley Garrett, who's here, spending her very last spring break as an undergrad doing field work in Alaska, where the temperatures were in the negatives in the morning and about 10 degrees Fahrenheit during the afternoon. And so we collected about 3 meters of core from both sites, and we're able to characterize those sediments and come with some of these, some data. And so I'm showing just some of the data, not all of it for the sake of time. But in both of these plots, on the left, we have Smith Lake 1, on the right, Smith Lake 4. In both plots, we show kind of where the active layer is and then where the stable permafrost is in the depth trend. And then I'm showing the median grain size, the D50, organic carbon content, and the delta C13 values of that organic carbon. And so as we kind of look at these plots, both sites, the sediment size, we have well-sorted silts, and the median grain size is about 35 microns. At both sites, we have low organic carbon in the permafrost itself. However, at site 4, we have our active layer where we have organic carbon values upwards towards 40%, 40 weight percent. However, I do want to point out that we're really, in terms of our global climate models and changing the carbon cycle, we're really interested in this deeper carbon that's been buried for tens of thousands of years, as this top stuff is still part of our current carbon cycle. But at both sites, we actually have a low point in TOC values or organic carbon values kind of at the top of the permafrost, and they increase towards the bottom of the permafrost profiles, and they get upwards of 2 weight percent organic carbon. And the delta C13 values of that organic carbon are about negative 25 to negative 26 per mil, and this is very standard for carbon or C3 plants. It's a very normal value. And yes, our deeper carbon is what's of interest here. And so this is kind of a small summary of what we've kind of been doing to classify these sediments. Now, I want to jump into trying to understand the gas flow in these permafrost sediments. So our main goal here was to gather flow terms for our modeling effort. And so in part of doing that, our first goal was to measure the effective permeability of gas in synthetic permafrost sediments as a function of ice concentration. And so what I mean by synthetic permafrost is a lab-made sample. And so this has been a lot of fun to use. And so if we think about permeability as a function of ice content in permafrost, we could use this little diagram to the right where we have sediments in brown, water in blue, ice in the light blue, gas in the red, and flow going through those pores. And so on the left, we have a sample that's totally saturated with gas. So our saturation of gas is 1, and we're flowing gas through that sample. And so we would call this an intrinsic permeability. So it's the maximum permeability that this gas phase can have because it's the only phase present. And then to the right, we have our sample with ice in the pore space. And so here, maybe the saturation of gas is down to about 0.5. And so you could theoretically think that, well, the permeability in this sample should be lower than the permeability in the fully saturated of gas sample. And so we would call this type of permeability effective permeability. So that's, it's explanatory, but it's the effective permeability, what's actually being measured there. And then if we take our effective permeability and we divide it by this intrinsic permeability, we get a parameter called relative permeability, or it's essentially a fraction. What fraction of our permeability do we have of the total permeability that's there? And so as we were designing these experiments, we thought using synthetic permafrost would be nice because then we can alter the amount of ice in our sediments, and that's the only variable that's changing. Because if we tried to look at this in our permafrost samples, well, we might have a grain size change, or we might have a mineralogy change. And so we came to the conclusion we're going to use a synthetic permafrost for this part. But in the future, we'll be measuring our permafrost permeability as well. And so to create a synthetic permafrost sample, we used a method called the undercompaction method. And this method essentially helps us to pack a sediment sample where we know the porosity and we can admix a known concentration of water. And so then we'll have a known water saturation of that sample. So in practice, this is what it looks like. We'll take our sample, we'll weigh it, we'll mix it with the known amount of water, and we'll split it into a number of sections. And then we'll take those sections and pack them into a latex sleeve that we have surrounded by a metal mold to hold the sample in place and confine it. And then we'll be able to pack our sections into our mold. And so there's a schematic of this to the right. Basically, we have some kind of bottom cap down at the bottom, and then we'll load in one of our packets of sand, pack it to the right height, and then do it over and over and over and over again until we have a sample. And our samples tended to be around 8 centimeters in height or in length. However, when we did this, we ended up noticing we had some problems with our water and ice saturation once we froze the sample from the top down to the bottom. We realized that the bottom of our sample had much more water than the top of our samples. And so this is just due to gravity. And so we actually found that even before we froze it, this water distribution had occurred. And so we had to think of a method that we could use to try and build a synthetic permafrost sample that didn't let the water drain to the bottom. And so what we ended up deciding to do is we took this freezing cap and we inverted it in our lab. And we have coolant lines running from a chiller. They go up through this brass cap, and the chiller runs this coolant at like -8 degrees Celsius. And then we can put our latex membrane over this cap, put our mold around it, and then start to pack our sample. But what we'll do is we'll pack one layer and let it freeze, and then we'll pack the second layer and let it freeze. And when we do that, we can end up with a sample with homogeneous ice distribution. And so just as an example of the before and after, over here on the left, we have a sample that's created from our old method where we just built the whole sample and then tried to freeze it, and our new method over to the right. And so this plot here in the middle is showing our sample height from the bottom to the top and the saturation of the sample as a function of that height. So our sample over here on the left, that's our old method, you can notice the top of it has a really light color at the top and a dark color down at the bottom. So this dark color corresponds to a really high saturation of water, and our light color corresponds to a really low saturation of water. However, our new method, we've got this nice homogeneous sample, and here we have a nice homogeneous distribution of ice. And so we were finally really satisfied with being able to make a synthetic sample. And in all, we measured the permeability of quite a few of these, and they just didn't plot how we thought they should be plotting until we realized what our mistake was. And so this actually took, months and months of refinement before we were able to kind of get it how we wanted it. Okay, and so once we had those samples, we could measure the permeability of those samples. And so this is how we do that. So over here on the left, we have our sample loaded into this cell. And so our sample's here, and it's got some porous stones on top and bottom. And then we can use this bottom port and fill up our cell around our sample with a confining fluid, and then we can pressurize that fluid so that our sample is kind of under the same pressures that it would be out in nature. And then we can use these ports down at the bottom with these red handles. And so these two are connected. One measures the pressure at the bottom of the sample, and the other one flows in our gas. So we're using nitrogen. And then it flows up through the sample, up through these lines, and comes out the top of the sample and out this downstream line. And then our pressure at the top of the sample is read at this other handle. And so this is kind of how we're measuring the permeability. And then this is our lab setup where we have our cell, we have gas, flows in through the cell, up through the sample, out, and then the flow rate gets measured here, and we have a computer to kind of gather all of our data. And the most important part, since we have frozen samples, our cell here is going down in this chest freezer where we're keeping the sample at around -2 to -4 degrees Celsius. And then, of course, we use Darcy's equation to solve for permeability. And so after this kind of painstaking effort, we finally have some data that we're happy with and we think is representative of what's happening in our samples. And so this plot is showing effective permeability on the y-axis on a log scale and the saturation of ice, and then a tiny bit of liquid water on the x-axis. And so essentially this is the saturation of 1 minus the gas saturation. And we can see going from our intrinsic permeability, or our permeability at a saturation of 0, we actually, as we increase the ice saturation, our permeability of our sediments is decreasing. And so we have over an order of magnitude decrease in permeability with an increase of saturation up to about 0.65. And so this was really nice to finally see after months and months of effort. And so we can also plot the relative permeability of these samples. And so if you remember, our relative permeability is the effective permeability, or what we measure, divided by the permeability of our sample at a 0 saturation. And so this plot to the right is showing our relative permeability on our y-axis and our saturation of ice on the x-axis. And we can see that our sample goes from relative permeability of 1 all the way down to about 0.06 with a ice saturation of about 65%. And so we can also fit this line with a Brooks and Corey fit of relative permeability, and we can model this data. And so this has been really exciting recently to get this data, and essentially this data will be used to inform our modeling effort. And in the very near future, we're going to be taking our core samples, cutting them, and loading them into our cell so we can measure their permeability as well. And then we can correlate our permeabilities with our other sediment characterization properties that we've measured. And then the challenge here is being able to cut the sample without melting it. And so every time we've tried to cut a piece of our permafrost, we just created like a nice new ice sheet. And so we can't measure the permeability if we melt the two ends. And so this might be a little more difficult than it seems, and then try to load that into our cell without melting the permafrost. So that's kind of where we're going in the very near future with this part of the project. Okay, so and to kind of wrap this up with what we're trying to do in our modeling efforts, we've got our third part here is our permafrost gas production. And so all of this data that you're about to see is Riley Garrett's data that she collected. And so obviously we want to make sure she gets the credit for this. So our goals here are to gather source terms for our modeling, and we want to perform incubations at current and future in situ temperatures. If you go through a lot of the literature for incubations of permafrost sediments, you'll find that most of these incubations took place at 25 degrees Celsius. And these soils are probably never going to get to 25 degrees Celsius. And so we want to try and see what the actual rates of production are at actual in situ temperatures and how those rates might change with changes in temperature. So if we have a sediment core, an example right here, where we have, samples located down through that core, and at different, at our current time, maybe those in situ temperatures are as listed here. And in the future, perhaps in 50 years at time 2, these are the new in situ temperatures. So we want to know how are our rates of greenhouse gas production changing from time 1 to time 2, even though it's over these very small increments in temperature. And so, yeah, essentially we want to measure CO2 and methane production rates, determine the effect of temperature on the production rates, and then we'll also utilize the carbon isotopic character of the CO2 and methane to try and backtrack and see what kind of microbial metabolisms we might be encountering in our sediments. And I decided for time to take that out, although I may have had time, but if you want to know, we'll talk about them later. And so this is how we're performing these incubations, and we're doing this in collaboration with Dan Breaker's lab, and he's helped us quite a bit with how to do the incubations and being able to measure the headspace gases from our incubations. So essentially, we start out with a glass vial and a lid that has a rubber septa on it at the top where you can poke a needle through that piece of rubber, and then when you remove it, it still stays sealed. And then we've taken 2 samples, one from each site, and so to incubate. And for those incubations, we took from Smith Lake 1, from one stratigraphic horizon, we are incubating those at 4 degrees C, so refrigerated, and at 25 degrees C. And then we did both of those in triplicate, so we can average the values together. And then we did the same thing at Smith Lake 4. And so what we do is we'll load our sample into our vial, and then we'll poke a needle down through there and we'll flush the headspace with nitrogen. And once we flush the headspace with nitrogen, we have an atmosphere in our vial that's totally anaerobic, where there's no oxygen or CO2 present, or methane. And then we're able to start our incubations. And so we did incubations for 15 days, 35 days, and 59 days. And then we collected the headspace gases on those increments of time. And once we had our headspace gas, we're able to measure the CO2 concentrations in a gas chromatograph mass spectrometer. And so in the future, we're working with Dan and his lab to outfit this GCMS to be able to measure CO2 and methane at the same time. And currently, we're only being able to measure CO2 at this point. And so this is actually something we're working on in probably the next 2 or 3 weeks to try and update the system so we can measure both, because we really are interested in the methane production, as methane's a much more potent greenhouse gas. Okay, so what we've seen so far with our incubation experiments is that CO2 concentrations are increasing with temperature and time. So just for reference, at Smith Lake 1, our sample comes from about 1.2 meter depth, and the organic carbon in this depth is actually really, really lean, 0.33 to 0.45 weight percent. And our Smith Lake 4 sample is from about 2 meters depth, and the organic content is about 0.8 to 1 weight percent total organic carbon. And so our plot here, we're showing CO2 concentration in nanomoles per gram of total material. And so this total material is the water and the sediment. And so we'll be readjusting these values to account for just the dry sediment, but the samples are still incubating. And so we can't burn off all the dry sediment yet, or all the water yet, I mean. And so on the left side, we have our Smith Lake samples. And so these clusters show our incubations at 15 days, 35 days, and 59 days for Smith Lake 1 at 4 degrees C, Smith Lake 1, 25, and then Smith Lake 4, et cetera. And so what we can see from this data is that we're having an increase in CO2 concentrations when we go from 4 degrees C to 25 degrees C. And we see that at both sites. And so to us, this was like really, really great news, because these were our first incubations that we've done. And so we were pretty happy about it. And then we can also see our incubations as a function of time at each temperature are also increasing. So we're not getting weird results as in, 15 days incubation, we have more than at 59 days incubation, right? And so all of this data is saying to us that, we're on the right track here, right? And so from here, we can move forward with more incubations at different temperatures. And so we're really happy with this data. And then most importantly are the rates of CO2 production. And so we can see that the rates of CO2 production are also increasing as a function of temperature at both of our sites. So this plot's showing CO2 production rate in nanomoles per gram per day. And so on the left, we have Smith Lake 1, on the right, Smith Lake 4. And so at Smith Lake 1, we're increasing our production rate as a function of temperature, and at Smith Lake 4, we're increasing our production rate as a function of temperature. And so this is really important because it kind of shows that what we're trying to look for and trying to pin down the kinetics of these reactions as a function of temperature or maybe organic carbon content is that we're finding that we can probably do this. And so this is really exciting data that I think we gathered it like last week. And so we're still really happy about it. Yeah. And so this is obviously going to be used, will be one of our most important source terms in our modeling effort to try and predict how much greenhouse gas we're going to be able to produce in our soils and then emit into the atmosphere. And so just our progress to date, we've made really great progress defining our sediment characteristics and our permeability behavior as a function of ice content. We've also made great progress performing incubations and gathering source terms of greenhouse gas production rates for our modeling. And of course, again, our data, our flow and source data are going to be utilized as inputs to feed into global climate models, most likely to be modeled by our friends at Sandia. Yeah. And so this is kind of where we're going and how we're trying to get there. And so that's all I've got for you today. Thanks for coming, and I'll leave you with a photo of our field work in Alaska, which was fairly low key. You kind of show up when we woke up and, you know, drilled with a little jackhammer, and compared to our UT GOM squared expedition in the Gulf of Mexico, which cost millions of dollars. And here's the drill string. And obviously it was 95 degrees every day out in the Gulf. So yeah, that's all I've got. Thanks.
All right, Tanner, thank you very much. And we are open for questions.
Thank you very much. That was a great talk. I do feel like you've left me on a cliffhanger. I was really ready to know what the permeability of your actual samples were.
Oh.
Okay, so I have a lot of questions. I think I'm going to kind of sneak into the first is for your synthetic permafrost. I was wondering what if you were using like uniform grain size for that and what that was, and if you looked at like how the grain size might affect the permeability. And then also I'm really interested in like the relic subsea permafrost. So I was wondering if you thought about like how the intrusive seawater at the top of the permafrost would maybe affect all of this.
Okay, cool. Yeah, so actually the second question, Kayla is working on a project for that right now, where she's looking at permafrost where the ocean is transgressed over that permafrost. And actually, yes, so like at the seafloor, that permafrost is being degraded. Yeah. And so it's starting to melt because of the water that's come up over top of it. Yeah. And then in terms of the synthetic permafrost, so right now we're using it's like a medium to fine sand. And so it's actually like pretty large grain size, especially compared to our sediments that we've obtained from Alaska, which are really silty. And so essentially we started using that for one reason, is that we had it and it was really it was there in our lab. But we actually started off with an even finer grained silt than our samples. It's called minusil. And we just had a really, really hard time working water into it at all and getting any kind of measurements. And so one thing we're actually going to do is we're going to do synthetic permafrost samples with permafrost sediments. And so when we were in Alaska, we went into a permafrost tunnel. There was an older guy that kind of has a personal tunnel that goes down through the permafrost, but he's like gold prospecting down lower. And he let us in his tunnel, and the people from Sandia were like, we're not allowed to go in there. Anyways, so we went down in there. It was really cool. But when we were probably 15 meters below the surface, and we actually grabbed a giant chunk of permafrost and brought it back to the lab, and I sieved out all the sticks and stuff. And so it's just it's like much more organic rich than our course. It's like 5 weight percent TOC. But we've got that sediment, and we'll be doing the synthetic style with those sediments. So it'll be like a true fake sediment. But yes, I do think like the grain size will affect the permeability and the rate of permeability loss with the ice, I think. Yeah.
Not very good at throwing pictures.
All right. I'm curious, in your synthetic one, do you think you've got, is it actually microbe free? So have you created an experiment that's truly in our, no life versus the life role between the two? And then I have a follow-up.
Yeah, good question. So I mean, there might be microbes in there, but not like at a large scale. So we're not like culturing bacteria and putting them into the sediments. So we're not producing gases from the sediments themselves, at least at this point. One of our colleagues at Sandia says he has like an extremophile bug that he wants to do some experiments with, but that's like, down the line.
Yeah, I guess the follow-up question then would be, could you have the ability, either maybe empirically, to quantify the amount of, say, water release and gas release from a microbe-rich or microbe-poor? Could you get to that point of saying, when it's a very rich microbe setting, it speeds it up because we have the water, and then the water refreezes, and then it changes the permeability in either a positive or negative route, versus one that has less life in it. With this ability to have a control.
So are you asking like, could we theoretically build a sample, put microbes in it, and then change whether we're thawing or freezing?
Yeah, I guess I was thinking about the metabolic reactions, for instance, that involve where water is one of the products. And that water then is part of the freezing component, which then you think there's a cycle back in again. And if you could say either the amount of gas changes because of that component, or the creation or loss of permafrost changes, could you actually get to that?
I don't know. That might be very difficult to. I don't know if the microbes would produce a ton of water to really change that. They might produce enough gas to like get beyond the saturation boundary, maybe. So it would maybe displace the water, I guess. But it's a very interesting idea. But I don't totally know.
So are you going to measure the methane concentration too? Because not only the amount of the greenhouse gas, but also the composition, the relative fraction of CO2 and the methane. That's an important factor.
Yeah, in the incubations.
Yes.
Yeah, we are definitely going to do that. Yeah. So Riley's going to be working with Dan Breaker to outfit their GCMS to measure methane and CO2. So the process of how this happens is the GCMS will sample the gas. It'll convey that gas into a wire, a tube, a metal tube. And then it actually lowers that tube into liquid nitrogen, and it will freeze the CO2 phase, and then everything else that gets conveyed out. And so that's how we collect the CO2 itself. And so essentially what we need to do is set up a second trap where we take those gases that include methane and we react them. Was he going to ignite them? Was he going to ignite them or just react them with oxygen and convert that methane back to CO2? And then we'll be able to measure the CO2 and the methane. Did I get it? Yeah? Okay. Yeah. Yeah, so essentially we need to set up a second liquid nitrogen bath to capture the methane that we turn to CO2. Yeah. And then the interesting thing there is we'll also be measuring the isotopes of the carbon from the CO2 and the isotope of the carbon from the methane. And when you do the delta C13 from the methane, it's really indicative of what type of methanogens you have in your system. So if you have a certain delta C13 value of like -50, that might be acetoclastic methanogens, but if it's negative 70 or 80, it might be the hydrogen-trophic methanogens. And so that'll kind of help us pin down what kind of microbes are there. But yes, so we're going to measure CO2 and methane. And we think it's going to be kind of a 1-to-1 ratio just based off of the stoichiometry of some of those reactions. But in practice, we'll see what we actually get. Yeah. And actually, I think it's the next slide. This is the Delta C13 data, right? But essentially, all of our data is plotting at more positive values than the parent carbon material. And so Dan thinks that this might be because all of the lighter isotopes are being put into methane rather than CO2. And so it's making our CO2 heavier or more positive than our parent value. But yeah, so it'll be really interesting to kind of look at that and measure both, yeah, and see the rates of both. Yeah.
Tanner, I am intrigued by this thallic layer, the unfrozen water.
Yeah.
So why does it form?
Yeah, so good question. So the way I think of it myself, so it's not always present. So sometimes you'll have an active layer just with permafrost under it. And so in the summer, you'll thaw down to a certain depth, and then in the winter you'll freeze down to there. But once this thallic starts to form, I think it's kind of showing that your potential to thaw in the summer is now greater than the potential to freeze in the winter, right? So the summers are bringing that thaw depth down lower, but then in the winter, it's not coming down as far. And so then it's leaving this layer that's totally unfrozen year round. Yeah. Yeah. And it's kind of the same process. There's a lot of lakes. Most of the lakes up in Alaska are kind of slump processes when this happens. And so there will be really ice-rich permafrost and it will like rapidly thaw and you'll actually get a big slump and it will leave a lake. Yeah, and so they're called thermokarst lakes. Yeah. And under all of those lakes, there's an unfrozen bit, yeah, because there's liquid water right on top of it.
All right, I think we might be wrapped. Oh, Alejandro, I have one more question.
A burning question. So in your sketch of the pore level, you drew the gas as the intermediate wetting phase. It's my understanding.
Yeah.
Is there any evidence for that, or like how did you come up with that?
I don't think there's any evidence. It was just kind of a quick schematic to kind of show what might be going on. But yeah, Peter said if there's any petroleum engineers here, they're going to get upset, which is what happened. Yes.
I might just close it. At some level, this is really blue sky research, which Tanner wrote a proposal for and was funded because it's very different. But in another way, many of the skills, at least some of us who are involved in the ocean drilling program, actually are a real strength in this kind of environment. Systematic characterization, also the whole process of coring and even sample archiving and thinking about how to do it. Those of us from the ocean drilling community actually have a lot to offer in this environment, even though it's a profoundly different environment. So it's been really fun for us, and Tanner and Riley have been really exploring new directions for our group. And I'll just remind you, Alejandro is gonna lead a lunch if anybody wants to join. I won't be able to go because I have to get back to campus. Tanner, thank you very much.
Thanks.